to another episode of Inside the Sciences. I am your host, Ishina Warhal. And I'd like you to imagine this, a life-saving cell therapy that is months in the making and it's in its final moment before being filled into vials, or a batch of, say, critical vaccines that is destined for thousands of people and is undergoing its final sterility testing. Now, the success of these moments and the safety of the patients hinges on one very important non-negotiable factor: absolute sterility. And so the modern solution is precision engineering, not just procedural control. So in this episode, we are going to take a look at the advanced technologies that are designed to actively create and guarantee, sterile environment. And these are called aseptic isolators. And we are joined today by Iza Al-Mokmina, who is a product management executive here at Esco, to help us, to walk us through these kinds of technology. Hi, Iza. Welcome to Inside the Sciences.
Hello. Everyone. I am Iza. So I'm the product management executive in Esco Healthcare. Thank you for having me.
Yeah, okay. Welcome to the show. All right. So let's start. We'll start with the core principle. So I'd like you to, because we have what we call aseptic conditions, and then we also have the containment, requirements. So what is the difference when we say aseptic conditions and containment in terms of, say, pressure and the purpose?
Okay. So first thing first, thank you for the questions. By the definition, aseptic and containment is totally different but related to each other.
Mm-hmm.
So by the definition, aseptic is the action of getting rid of the microbial contamination
Mm-hmm.
in a manner that requires a high level of cleanliness in a certain area. For example, before a pharmacist reconstitute an IV injection inside a unidirectional airflow cabinet. The pharmacist should wipe first the working station, and even the vials itself, it should be swabbed with alcohol or other disinfectant, usually ethanol or isopropyl alcohol.
Mm-hmm.
And ideally, that aseptic condition is already achieved where the working station and the vial are swabbed with the disinfectant agents.
Mm-hmm.
So that's the example of the aseptic. And while for the containment itself, it's a system or method to prevent any substance, any outside particulates or unacceptable materials from infiltrating into a containment system.
Mm-hmm.
For example, the isolator chamber. Or in another way, to prevent any materials inside the containment system from going outside or breaking out into the surrounding area.
Mm-hmm.
And if we proceed further to the isolator, an aseptic condition is required to maintain the sterile environment achieved through the use of positive pressure. For example, again, to decontaminating the chamber with vaporized hydrogen peroxide. So it simply implies the taking action to achieve the aseptic condition. And while for containment, in the isolator, for example, to maintain the aseptic condition inside the isolator. So the function of the isolator to isolate the materials inside the isolator for not going outward from the isolator chamber. So it's containment.
Okay, so you mentioned about pressure, negative and then positive pressure. Can you tell us the difference between the two? Like, say, for example, if we use one of Esco's products, GPPI, what do you mean when you say negative pressure and then positive pressure?
Actually, GPPI or General Processing Platform Isolator has either option of positive or negative pressure. But if you commonly find that GPPI has the positive pressure, it's simply because the pharmaceutical industry required as it is. And one common procedure or testing that pharmaceutical industry usually perform is sterility testing. So by the CGMP, or Current Good Manufacturing Practice, by the PICS, sterility testing is mandatorily performed under the aseptic condition.
Okay.
Yeah, so that's why in GPPI, it has the positive pressure, which will prevent the particles from outside the isolator from going inward to the isolator. So the air will flow toward the outside environment, thus ensuring the aseptic conditions. And once there is an option of negative pressure, then yes, I have mentioned earlier that actually the negative pressure is representing the containment definition. So usually it's used by the containment system, as the air will flow inward to the isolator chamber, thus preventing the hazardous drugs inside the chamber from contaminating the environment.
Okay, all right. So, how about if, say, for example, there is a manager and that manager is assessing their needs, so what are the key questions that they have to ask so that they can find out whether they need aseptic conditions, they need containment protection, or both?
Okay. As in the common request from the users or our customers that requires for an aseptic condition or containment protection. Yeah, so actually those are different definition or different process. So it will be has its own solution. So for example, for the aseptic conditions. So for the aseptic conditions, the manager or the user must assess what actually their process is. For example, they require to do the sterility testing. So it requires the aseptic condition. So that's why they will need an isolator that will provide the aseptic condition or unidirectional airflow.
Okay.
And other application, for example, are filling line of drugs vials in mass production or smaller production, or drug clinical studies, sterile compounding like total parenteral nutrition, or preparing intravenous injection. And another thing, cell engine therapy, for example, like vaccines production, antibody monoclonal production or handling, and etc. Meanwhile, for the containment requirements, the managers should assess whether the substance that they handle is hazardous or not. Hazardous powder or highly potent API or active pharmaceutical ingredients, or not at all. So yeah, if it's hazardous, if it's highly potency of drugs, so they need a containment solution of isolator. Which I have mentioned, need to use the negative pressure isolator.
Yeah, okay.
And other application maybe like dispensing, milling, or formulation and sampling for QC for also the hazardous drugs.
All right, so those are really important things that we have to remember. Okay, so now let's dive into the core technologies when it comes to aseptic and containment models. Let's talk about biodecontamination, because as you've mentioned earlier about one model here in Esco, the GPPI, right? And GPPI, as from what I understand, is fully integrated with hydrogen peroxide system, right? So can you tell us, why is it that an integrated automatic cycle such as the GPPI is more reliable in achieving, sterility?
Okay, it's also a good point for the biodecontamination system. So yeah, as per the CGMP again by the ICS, to handle or to produce a sterile product under aseptic conditions, environmental monitoring should be done. So there are two main points of the environmental monitoring that are usually performed under the isolator. First is the particle count monitoring. So since it's an aseptic and sterile environment, um, it's indicated by the grade A cleanliness. So grade A cleanliness is the highest level of grade cleanliness of the room. And the second is the microbiological monitoring. So to prove that the sterility of an isolator after the chamber decontamination is maintained. And hydrogen peroxide, as you have mentioned, is one of the safest disinfectant for the environment and humans. When it breaks down into H2O or water and O2 or oxygen in the air. Hence, it becomes common in isolator for the pharmaceutical industry to use the H2O2 or hydrogen peroxide for biodecontamination. And the efficiency in achieving the sterility assurance level or SAL is proven very well of the 6-log reduction, which means only 1 per 1 million microbes remain alive.
All right.
And you mentioned that why it is more reliable with the integration of the biodecontamination. So actually by integrating it to the isolator, that what makes it reliable, more reliable and practical for the user to use the isolator instead of separate equipment or mobile biodecontamination. It's very, very it's better to have the dedicated to the isolator itself, where the validation of the decontamination development was performed successfully and able to give the automated cycle of the biodecontamination upon daily use. Thus, it will be easier for the user to operate the isolator instead of having the separate biodecontamination equipment, because it requires the operator or the user to revalidate again the mobile biodecontamination, or it's like I'm having the different recipe for this certain isolator. Maybe isolator A have this recipe A and isolator B has recipe B. So it's like with one only mobile biodecontamination, the user must switch in each single time they do the biodecontamination for the isolators.
Yeah, I get it now. I see the practicality of it.
Correct.
All right. So, speaking of still GPPI, so from what I understand also, GPPI offers a choice between a single pass and then a recirculating airflow. So can you walk us through a specific scenario for each? For example, when you're handling potent, say, compounds versus non-potent materials.
Okay, yeah. I will also take an example of GPPI again. So yeah, GPPI has actually the on-site configurable airflow regime. So the user can choose in the field whether they wanted to use the the single pass or the recirculating airflow.
Yeah.
So I will start first with the single pass, or known as total exhaust. So if the processes requires the direct flow out of the air, out of the room or the building, so it's like the air must go outward outside the building, so surround the environment by linking the ducting collar from the isolator to the HVAC system. So HVAC is heat, ventilation, and air conditioning of the building from the ducting pipeline of the building. And actually, single pass system is used if the process involves the volatile and corrosive solvents and even other hazardous substances that can harm humans.
All right.
While for the recirculating itself, actually it's quite simple and quite practical. It involves relatively safe substances and process. So the exhaust air to the room is allowed and doesn't harm humans in the surrounding area.
Okay, all right. So let's now shift to containment. So we have what we call the weighing and dispensing containment isolator, or the WDC, right? So, and we also have the CBIT which handle potent powders. So how does turbulent airflow in a negative pressure system safely contain such hazardous particles?
Okay. Yeah, so now we're talking about the containment isolator.
Mm-hmm.
So when handling the potent powders, the turbulent airflow is actually used to spread powders to all over the isolator chamber.
Mm-hmm.
Thus, it makes a it creates a low level concentration of the powder. Thus, it's not suspended to only one spot inside the isolator with a high level concentration, and it will makes difficult to wash down the isolator chamber.
Mm-hmm.
So yeah, in WDCI and CBIT, it is common to have the accessories or features of a spray gun or spray ball, and the function is to wash down the isolator chamber. So this spray gun or spray ball can spray the purified water or compressed air to clean the chamber from the suspended highly potent powders.
Okay, and we also have the turbulent flow aseptic isolator, or the TFAI, right? So why does it have an ISO class 5 conditions?
Okay, yeah, that's a good point for a turbulent airflow, because usually turbulent flow in isolator is only used for the non-sterile application. So yeah, so in this device, so it's very highly customizable and specific, where this device has the purpose of handling the potent powders with its turbulent flow. And what differs T5 from WDCI and CBIT is the aseptic condition, or ISO class 5, that is given by the integration of the biodecontamination system. And usually T5 or TFII is chosen if the user still requires the aseptic condition when handling maybe a small amount of potent powders.
Okay, all right. So, speaking of WDCI, a key spec of containment is what we call the leak tightness, right? So, and WDCI is tested to ISO 10648 2 class 2. So, why is this quantified leak rate so important when it comes to risk assessment and operator safety?
Yeah. Okay, yeah. Before that topic, actually, I would like to recall again the definition of an isolator that was explained by Handini.
Yeah, in the previous
in the
podcast.
previous section. So when she was differentiating the wraps and the isolator. So yeah, isolator with its fully sealed design, whether with static or inflatable seals, and its pressure tested unit according to the ISO 10648-2, which explains the allowable hourly leak rate of an isolator from class 4, the low level, up to the strictest, which is class 1. So yeah, by this regulation, the isolator is used to contain the process or the application, so as not to be contaminated by the outside environment. So that's why the pressure test for isolator is mandatory to ensure that all the corner or the gaps from the isolator or equipment is tightly closed. So that's why this pressure test is mandatorily required for either aseptic isolator or containment isolator.
All right, okay, that's clear. Now let's talk about, let's highlight some different models that we have here in Esco. So we have the cell processing isolator, or the CPI, right? So this is described as an integrated system, and it's not just an isolator. So can you give us an example of how it combines equipment in order for it to create a seamless, sterile workflow, particularly for cell therapy? And why is it that integration is quite critical for product quality?
Okay. So CPI, or cell processing isolator, is actually a unidirectional airflow isolator, which is dedicated for the cell processing application.
Mm-hmm.
So CPI can be integrated mechanically with the incubator, centrifuge, refrigerator, microscope, etc. And for example, where the process is using the incubator, and the user needs the incubator to be located next to the isolator, because they will take the incubated cell after incubated in the incubator directly under the aseptic isolator. So that's why it will be requiring the aseptic conditions.
Okay, and how about for fill finish operations? From what I understand, Esco partners with machine manufacturers. So how does this collaborative approach to building, say, a filling line isolator result in a more robust and compliant solution than, say, a generic off-the-shelf isolator?
Okay. Yeah, we have also the filling line isolator, which is very highly customizable isolator. And we also have the partnership with the third party of the filling line machine manufacturer. So basically it involves the compliance with the user's requirements to build a process dedicated machine to fit into the Esco unidirectional airflow aseptic isolator, which is the filling line isolator. And certainly, it will give a totally robust and complete design of the filling line machine, as the user doesn't need to adjust here and there, which will burden them to have a double commissioning or validating the complex equipment.
Yeah, I understand that. It's really that would be tedious. Okay, so for example, we have potent powder and we have to handle this potent powder, and you have the WDCI and then you have the CBIT. So what are the differentiating factors that would make a client actually choose one over the other for, say, weighing and dispensing application?
Okay, yeah, actually by the basic principle, they are used for the same way, which is to contain the potent powders or hazardous drugs. So what differentiate them is firstly the seal of the door. So yeah, it's basically based on the design, the mechanical design. The seal of the door and the dampers. So WDCI features an inflatable seals that is supported by the compressed air, allowing for deflation. So it will open the isolator, and inflation, so it will close tightly the isolator opening door. And secondly, the valuable integration system from the WDCI is the integration like an audit trail. So this audit trail is complying to the 21 CFR Part 11 regulation. And WDCI also has larger display size on the touch screen human machine interface, or HMI, than the CBIT. And WDCI has actually the high-end design, which allows the user to have highly integration or easily configurable options. For example, integrate the drum lifter, platform with stairs, or even the explosion rated components, and etc.
Okay, so now with all that, I think let's now go to the features that make these modern isolators. We could say they're intelligent partners when it comes to regulatory compliance and process validation. So we all know that data integrity is really quite, it's important. So how do features say we have GPPI. GPPI has this optional 20 plus CFR Part 11 compliant data, so data logging. And also WDCI has this automated pressure decay test. So they not only capture data, but also they streamline the validation and audit process. So how do they do that?
Okay. The main point is traceability. So traceability in industry is one very important thing. Data and alarm log and audit trail to comply with the 21 CFR Part 11 are the added value features to the equipment. 21 CFR Part 11 is regulated by the Food and Drug Administration, or FDA, of the US, which implies that the electronic records and electronic signatures are recommended to be integrated in the control system of certain equipment. In this time we talk about the isolator. And 21 CFR Part 11 expects the equipment should have the ability to save accurately the data of the alarms, the monitoring parameters, for example, like the airflow velocity, like the filter differential pressure, and even the events that happen, like who the user that removes or delete or change the data alongside with the date and time. So it gives the compliance of the electronic records. Well, for the electronic signatures, it is equipped with the unique user ID and password for each user access level, for each operator, supervisor, admin, and maintenance account. It should have unique user ID and unique password. So overall, this regulation has the intent to prevent any manipulation of data and maintain the data integrity.
All right, that sounds really intelligent. Okay, so for my last question for you, Isa, if we look at the portfolio of Esco, the theme is customization, from the GPPI's 20 plus options to building a fully custom CPI. So my question is, how does design process ensure that isolator doesn't just fit the room but actually enhances the specific process that it's built on?
Okay, so in Esco Healthcare, specifically in Esco Pharma, so we ensure that Esco's recommendation solution to our valuable customer fulfills their process. So the communication and relationship with the customer should be maintained until we reach the deal of the agreed suitable equipment. And to reassure that customers certain to our unit, we provide the detailed drawing to help customers visualize their specific needs. And also prompt and knowledgeable discussion with the customers is our priority to ensure the process of earn the deal is smooth. And so finally we can build the very fit and suitable equipment for the customers.
All right, okay. So, Isa, I learned so much from you. Thank you very much for sharing your expertise with us today in Inside the Sciences. Thank you very much. Okay, that has been a very incredible dive into the isolation technology. So if you want to know more about, the full specifications of the models that we talked about here at this episode, go to www.escolifesciences.com. And once again, this is Sheena, and this has been Inside the Sciences. Bye!